The final lab that we'll review is called Relationship and Biodiversity. In this lab, we were given a plant called Botanocurus. Botanocurus is said to be endangered, so it's an endangered plant that produces curel. Curel is said to cure cancer. Because of its endangered status, we have to find an alternative plant to use. Alternative means a different plant to use. This lab will compare several species to see which is most closely related to Botanocurus. We will use both structural evidence and molecular evidence to compare the species. So whenever you're given the task to see if one species is related to another species, whether the species are a plant species or an animal species, we can use both structural and molecular evidence to compare. Um, you are aware of some of the techniques that we use today to compare one person to the next. For example, DNA test is one of the ways we could use, or we can also use a fingerprint test. We can also do an identity test where you're looking at people to see similarities. So we basically use these methods today, and you are aware of them, but we are going to look specifically at Botanocurus and see what species can be used since Botanocurus is an endangered species. I have to also remind you that endangerment means that there's a small amount of those species left in the wild. And since it provides a medicinal value, we have to protect the species. Also because of biodiversity and the balance of the ecosystem, we should protect the species. Um, However, we can't continue to use the species because it will lead to extinction, and we do not want that because that will end. That will be the end to all cure for cancer. Again, this is a hypothetical situation. Um, the regents really wanted you to have an experience dealing with something that, okay, it's on the brink of endangerment or extinction. It provides a medicinal value. What should we do, or what our next steps are in terms of protecting the species and finding an alternative source. Let's start off with our structural test. Now, structural tests, or the plant structure, is the physical plant. It's looking at the plant, placing the other plants that we're comparing side by side, and seeing or noting the similarities and things that are not so similar. So let's start off with Botanocurus. Let's look at the features that this plant have, or structure that this plant have. First of all, the plant has a flower, has leaves, a long stem. The flowers are purple and yellow. The leaves are curved at the edges. The other species, species X, have a flower also. The flower is yellow. The leaves are pointed at the edge. Species Y, it is blue. The flower is blue. The leaves are almost pointed towards the end also. And then species Z, it also have a mixture of purple and yellow. The leaves are also pointed. So the similarities are with the other plants is that all of them have flowers. However, species Z flower and Botanocurus flower are almost identical in terms of coloration. Their leaves, however, none of the leaves of the plants had anything similar to that with Botanocurus. So we already note our structural characteristics of the plant. If this was an animal, we could have done the same thing. How many legs? How many arms? How many eyes? Well, some animals have multiple or compound eyes versus simple eyes. So if you're ever wondering why I say how many eyes. Um, yeah, there's so many things that we can compare in terms of just looking at the plant or animal and comparing them. We can also compare in terms of plant the seed structure. So we're going to compare the seed structure, that is how the seed looks, compare that to the other species. So this seed is kind of elongated and is relatively small. Species X is brown, it is circular. Um, Species Y is small and elongated. Species Z is elongated and also it is relatively small. So the similarities are all of the seeds are small. The dissimilarities, or the things that are not so common in Botanicurus, is that species X and species Y have a different shape and color than that of Botanicurus. We can also look at the stem for plants to see whether they are similar or not. For Botanicurus, we have the vascular bundle is in a circular arrangement. That means that it's aligned around the edges of the plant. Species Z, it's also aligned around the edges of the plant, but species X and Y, they are more scattered. They have a scattered arrangement. So the similarities is that it have a very similar vascular bundle arrangement to species Z. Species X and Y, they're scattered, they're not so similar with Botanicurus. Now, let's do our molecular test. Now, molecular test is not something that we could just look at the plant or parts of the plant and determine whether they're similar or not. We have to do more molecular or have more molecular evidence. And the molecular evidence we could have comes from these tests. One of the tests is called our gel electrophoresis. Now, again, two things that you should know. How can we compare the molecules or what's the molecular evidence we could have when we're comparing species is that we can compare their DNA sizes. And the test that we use to compare the DNA size is called gel electrophoresis. electrophoresis. Electro meaning electric, phoresis. So we're basically passing electricity through the gel, and we are going to separate the DNA based on their size. So we have a well here, and we put our DNA sample for species Botanicurus, for species X, Y, and Z. We do the same. And then by running an electricity at this end, is going to separate the parts based on size. The smallest part will move the farthest, and the largest, heavier parts will stay closest to the well because it's harder to move. So the electricity will separate the size and it will form bands. If you look at Botanicurus and species Z, the size of the DNA are identical. However, with species X and Y, the sizes are much different. So again, one molecular test we could do is a gel electrophoresis test. What we're testing to see the size of the DNA. We can also do an enzyme test. An enzyme test is to test the plant for a specific enzyme. In this case, we were testing enzyme M. Botanicurus had a reaction with with a chemical, and it proved to have enzyme M. Species Z also had enzyme M, but X and Y, there was no reaction, no bubbling, no enzyme M. So you also saw this when we were doing the experiment in class. We can also separate the dyes of plant if we want to compare them. To separate the dye, we extract the dye by crushing the plant or a part of the plant. And by taking a pipette, we extract the dye and we drop it onto filter paper or strips of filter paper. These are all strips or filter paper. So by dropping the dye at one end, dipping it in some water and leaving it for like about 2 to 3 hours, the dyes will start to separate based on color. So the dyes that Botanicurus have were purple and yellow. The species Z also had purple and yellow, but the other species you could see they had blue and they had pink and all these other colors. So again, dye or paper chromatography is another way to compare plants and see if they're related or not. Chromatography is CHROMATOGRAPHY. Chromatography. Remember this word also because that is another test you could do, a chromatography test. So we're finished with all of our tests. Say for example, we have our DNA sequence for this plant. We have our DNA sequence for Botanicurus being CAC, GTG, GAC, TGA, GAA, GGA, CTC, CTC. Now that's our DNA strand. They want us to come up with the sequence of bases in the mRNA strand. Now there is a general rule whenever you're doing the sequence of an mRNA once you're given the DNA strand. And we're going to complete this chart using these rules. The rule is, for any RNA, every time you see a C in a DNA strand, you pair it up with G. Whenever you see G, you pair it up with C. Whenever there's an A in the DNA, you pair it up with U in RNA. Whenever there's T in DNA, you pair it up with A. So again, the rule is C pairs up with G, G pairs with C, A pairs with U, and whenever you see a T in DNA, it pairs up with A in RNA. If you notice the difference, if this is a DNA strand and this is an RNA strand, the DNA strand, DNA does not have U inside of it. RNA does not have T inside of it. Please make a note of this and remember, you will have to do this in your exam. C pairs with G, G pairs with C, A pairs with U, T pairs with A. Now let's go back to the chart and finish that up. So we're going to C pairs with G, A pairs with U, C pairs with G, G pairs with C, T pairs with A, G pairs with C, G pairs with C, A pairs with U, C pairs with G, T pairs with A, G pairs with C, A pairs with U, G pairs with C, G pairs with C, A pairs with U, C pairs with G, T pairs with A, C pairs with G, C pairs with G, T pairs with A, C pairs with G. So now that we have completed the RNA strand, so they give you the DNA, you have to come up with the RNA strand. And C pairs with G, G pairs with C, A pairs with U, and T pairs with A. Again, please remember this, it's very important. Another thing you'll have to do is you have to come up with the amino acid sequence. Now, to get the amino acid sequence, we're going to use a chart. The chart that we're going to use, they're going to give you this reference chart. And this is called the Universal Genetic Code Chart. Now, the first thing you do is that you note whatever, so here's a completed one. So they give you the DNA strand, you come up with the RNA, which we did already. You have to find the amino acid. So you look for GUG of the RNA in the chart, GUG. And GUG is found here, so the amino acid is VAL. And that's why they have the amino acid VAL. So here's the completed chart. The completed chart is that they give us the DNA strand, we come up with the mRNA strand, and then we use the chart to find our amino acid. Then we can now compare our amino acid sequence with that of Botanochuris. Now, if the amino acid sequence is the same, it means basically that the plants are similar. They will make the same proteins. If the amino acid sequence is different, even by one amino acid, it will mean that it will make another amino acid, and the plants are not related. They are probably related to some extent, but they're not so closely related that it will produce curel. So the sequence is, there's a val, his, lu, thr, pro, glu, glu. If we look at species Z, it is val, his, lu, thr, pro, glu, glu. So they have the same exact sequence. That means that it will make the same protein. There is SER in species Z and SER in species Y, which are not found in Botanochuris. So we can go ahead and say that species I'm sorry, Botanicurus and species Z is related because they have the same amino acid, and we can also say that species X is related to species Y because they have the same amino acid. So whenever we're comparing plants or even animals, we can use the DNA, the amino acid sequence to tell us whether they are related or not. So just a summary of this lab is that to compare species, we can use both structural and molecular evidence. Some of the structural evidence that we can use for plants or animal is the plant or animal structure, that is their physical description, how they look. We can compare their stems, we can compare their seeds, we can compare their animals, we can compare their bones. And all these are structural evidence, or things that we just look at and make a comparison. Molecular evidence are things that we could do, but we need to run some tests. We need to compare them on a molecular level. We can do DNA sequencing or gel electrophoresis tests to separate the DNA based on size. And that's also called a DNA test. We can do a fingerprinting test. So species that are if it's animals or people, if they're closely related, will have very similar fingerprint. We can do a dye comparison. A dye comparison is extract the dye from the plant, put it on a paper, and that's also called a chromatography test. And we can also compare the enzymes, and finally, which I don't have on this slide, but we can compare the amino acid sequence.